guys, my name is Vaishnavi. In this video, I'll be talking about cochlea and the organ of Corti. As we know, the internal ear has bony labyrinth, which houses a set of continuous fluid-filled epithelium-lined tubes that make up the smaller membranous labyrinth. Two divisions of the membranous labyrinth are vestibular and cochlear labyrinth. Cochlear labyrinth provides assistance for hearing and contains the cochlear duct connected to the saccule, which is highly specialized as a sound receptor. The cochlea contains three spaces: scala vestibuli, cochlear duct or scala media, and scala tympani. The cochlear duct contains endolymph and ends at the apex of the cochlea. The other two spaces contain perilymph and are one long tube beginning at the oval window and ending at the round window. Along its length, the cochlear duct is separated from the scala vestibuli by the vestibular membrane. It's a thin structure consisting of a basement membrane with simple squamous epithelium on each side, facing the cochlear duct from one side and scala vestibuli from the other. In the lateral wall of the cochlear duct is stria vascularis. It's a unique columnar epithelium responsible for production and maintenance of the endolymph for the entire membranous labyrinth. The stria vascularis encloses a network of capillaries and consists of cells with many deep basal infoldings of the plasma membranes. Fluid and potassium ions pumped from the capillaries by these epithelial cells are released in the cochlear duct as endolymph. Cell bodies of bipolar neurons in the spiral ganglion send dendrites to the hair cells of the spiral organ and axons to the cochlear nuclei of the CNS. Spiral organ or organ of Corti. It contains the special auditory receptors in the form of hair cells that respond to different sound frequencies. The spiral organ rests on a thick membrane, which is called the basilar membrane. There are two major types of hair cells: outer and inner hair cells. Outer hair cells have a curved row of long stereocilia, which are embedded in the tectorial membrane. And the inner hair cells have linear array of short stereocilia. No kinocilium is present on cochlear hair cells. Two major types of columnar supporting cells are pillar cells and phalangeal cells. Pillar cells outline a triangular tunnel-like space between the outer and inner hair cells. Phalangeal cells intimately associate with the hair cells and contribute to the tight epithelium, separating endolymph from the perilymph in the scala tympani. The stereocilia of cochlear hair cells detect movements of the spiral organ. Sound waves collected by the auricle of the external ear cause the tympanic membrane to vibrate, which causes movement of the ossicles in the middle ear, eventually leading to pressure waves within the perilymph beginning at the oval window, moving along the scala vestibuli and causing movements of the vestibular and basilar membranes and the endolymph surrounding the spiral organ. High frequency sound is maximal movement of the spiral organ nearest to the oval window. Lower frequency is when pressure waves move farther along the scala vestibuli, displacing the spiral organ farther from the oval window. Sounds of the lowest frequency that can be detected produce movements of the basilar membrane at the apex, the helicotrema of the cochlea. After crossing the cochlear duct and spiral organ, pressure waves are transferred to the scala tympani and exit the inner ear at the round window. The pressure waves produce movements within the spiral organ that cause the hair cells to depolarize and release neurotransmitters to afferents of the cochlear nerve, producing signals interpreted in the CNS as sounds. Thank you.